I'm pleased to introduce Professor Christopher Sweeney. He is a leading clinician, researcher specialising in the treatment and management of genitourinary cancers. I hope I've got that pronunciation right. Good. Particularly with prostate and testicular cancers. He is the inaugural director of the South Australian immunoGEnomics Cancer Institute and a professor of medicine at the University of Adelaide, as well as being a consulting medical oncologist at the Royal Adelaide Hospital. His work focuses on drug discovery, development and personalised medicine, with a strong translational approach that bridges laboratory research and clinical application. Professor Sweeney is also deeply involved in clinical trials aimed at improving the patient outcomes through innovative therapies. Today, Professor Sweeney will talk about turning cancer research into real impact. Thank you.
Thank you very much for this incredible opportunity to share what we are doing at Adelaide University, the merger of UniSA and University of Adelaide, just down the road in the AHMS building. What we mean by turning cancer research into real impact, we at SAiGENCI and the cancer research community at large at Adelaide, we're not here just to write discoveries, put them into papers. We want that data to generate new discoveries that we turn into new therapies and new ways to prevent cancer and treat cancer. So not only discover, we are setting up our structure to do something about our discoveries, making use of the Adelaide ecosystem. I'd first like to just point out that Adelaide has come a long way. Sorry, the pointer is here. Mark, you taught me, but I'm failing what you taught me. Like a true professor, maybe enter. There we go. So I'm representing an incredible team of great talent that has come into Adelaide. As you've seen, the precinct down on the west side has just grown dramatically, just like this Adelaide Oval. I remember being in the 1970s in Yobbo Hill, and there was no building around us. Clive Lloyd, Viv Richards, Dennis Liddy, and Jeff Marsh and the Chapman brothers were duking it out, and there was nothing there, just like the west end of Adelaide. Now this huge precinct, and frankly I think there was somewhat of a brain drain to South Australia, and in 2022-2023 we tried to actually bring in an influx of an incredible amount of new talent to add to the South Australian cancer research ecosystem, to what was at SAMRI and is still at SAMRI, where Susan Watts is, and what the University of SA had in the Centre for Cancer Biology. So with the 20 group leaders we've brought in, plus the teams at SAMRI and at UniSA, all under the Adelaide University umbrella, there are about 40 to 50 high-level functioning, globally recognised cancer researchers in Adelaide, which is quite an incredible number. But that's going to come together by the team that we've built and working together. And so Jose Polo is an expert in epigenetics, and Luke Isbell is in his team, and we'll discuss what that is. Lan Nguyen is computational systems oncology, which is basically the umbrella term for how to manage large volumes of data, be it genetic profiles, genetic mutations, or various ways of analysing biological data, but also how to analyse clinical data. And you've probably heard of the term artificial intelligence. So this is that type of research at scale in our home of Adelaide. Lisa Butler is a leader in trying to work out how to prevent resistance to therapy and leading a program called Resistance Prevention. And then Brendan Jenkins from Nord High School through the University of Adelaide, went to Monash and is now back in Adelaide as well. So an incredible team leading these 4 different programs which are cutting edge to help make a real impact. patient. There we go. So this is the west end of Adelaide. It used to be a skate park, if you remember it in the day. Had the Royal Adelaide Hospital go up, the SAMRI building go up, Adelaide Bragg Center. Yes, the proton is on its way. Don't know the exact details, but it's going to happen. Adelaide University, where in the AHMS building, and then the Bradley building, and then across the road is CMAX. And within all this ecosystem, we've also got the University of Adelaide premises as well, where we can make drugs with the chemical engineers and the chemists. Yeah, out at Walton Lakes, they have facilities as well. So we have the ecosystem not only to discover things, but also do something about it, turn it into drugs, turn it into devices and the like. So I would like to just have my team speak for me about them, because they do a better job in this video that we've created. So go video. SAiGENCI is a new cancer research institute nurtured by the University of Adelaide and the Central Adelaide Local Health Network, focused on improving the care of patients with cancer or at risk of getting cancer. We are designing new programs focusing on the leading edge and having new, talented, fresh, energetic investigators to deliver on those goals.
There is a whole field of cancer epigenetics where the idea is, okay, we cannot do anything about that mutation because it already happened, but we can learn how to control the epigenome. If we know how to control that epigenetic layer, we should be able to reprogram any cell type into any other cell type. If you can convert a cell of the skin into a blood cell, why you will not be able to convert a cancer cell into a no-cancer cell?
When I started in prostate cancer research, there were very, very few treatments for men once they had progressed and become resistant to the standard treatment. In the last 10 years or so, all of a sudden there's been a number of different agents that have been approved for prostate cancer, but unfortunately it still does remain incurable. And so that's really what's driving us, is trying to understand, why does everyone become resistant?
Half of all of cancers are linked to chronic inflammation. And so one of the visions that we have for our program is that we can develop new drugs that target the immune system and also suppress the cancer. And in doing so, we hope to improve the quality of life of millions of people, not only in Australia, but worldwide.
In the computational system oncology program, our hope is to be able to make something equivalent to weather forecasting model, but for cancer.
We want to find patients before they develop cancer and develop specific prevention or screening strategies.
One of the things that really excites me about SAiGENCI is the new interactions and collaborations that we can establish with neighbouring research institutes.
We have researchers from multiple disciplines working together in the same institute with one goal, which is improve outcome for cancer patients.
We can all achieve much more in an environment like that than we could in isolation.
We're going to develop and deliver better strategies to improve patient outcomes, not only in South Australia, but all around the world. So the other thing I would say is that it's not the older people who are going to make these changes, it's the next generation who are leading the charge. And one of the examples about emerging, actually I would say emerged, junior cancer research leaders is Luke Isbell, who's going to talk about this field of epigenetics. So thank you, Luke.
Thank you, Chris, for the introduction. And it really is a pleasure to be here. This is the best part of my job, sharing the science that I'm passionate about with passionate people. I absolutely love it. I will drill straight into what we're talking about today. I will explain what epigenetics is for those non-biologists out there. And I'll share with you a project that I'm particularly excited about that is going on in my lab for the next few years at least. So for those who haven't heard of epigenetics before, I think the best working model or example of it is standing right in front of you. You can ignore the slide and look at me at the moment. I'm an example of epigenetics in action. Now, we all know that each and every single one of our cells in our body contains the same DNA, and yet they are clearly distinct. Why is that? Well, if the DNA is the blueprint to life, it's not like we're reading all of those blueprints at the same time. You've got your heart cells reading and activating the genes that are important for your heart, for example, and you've got your brain cells reading and activating the genes that are important for your brain, okay? And we call this selective reading of the genome, the selective interpretation of our genetic material, epigenetics. And in this way, every single one of your cells in each and all of your bodies has an epigenetic identity, which makes that cell what it is, okay? Now, it's worth really focusing on this process of epigenetic identity because it's quite a dynamic situation. As we're developing within the uterus as an embryo, we start as a single cell with the same DNA, and that cell divides and it grows and it changes. We call it differentiation. And this is part and parcel with the gain of new epigenetic identities specific to each of the different tissue and cell types within our body. So this is, as I said, very dynamic as a process. It occurs all the way up to birth and then even continues beyond this gain of different cell types, different epigenetic identities. Now, we know that all diseases take place within the body and have their own cellular identity, which is in a way an epigenetic identity specific to that disease. Now, epigenetics doesn't cause every disease, but because every disease goes in the context of a body, epigenetics is working hand in hand with the identity of that disorder, be it developmental disorders or indeed cancer, where we know increasingly epigenetics takes a very big role in the establishment and development of that disease. So this is an emerging field, and it's critical that we understand epigenetics now for two main reasons. One is better prognostics. This is a new tool that gives us a way to look at cancer or developmental disorders through a different lens. And often being able to understand a disease is extremely valuable for the person with that disorder. And also, if we understand disease better through an epigenetic lens, we may find that we have new processes and strategies with which to target that disease. Now, I'm going to talk about a project we're taking forth with my lab, targeting a particular protein that has epigenetic function. It's called P53, and it's often referred to as the guardian of the genome. And that's because P53, in each and every single one of our bodies on a daily basis, prevents the cells in our bodies from getting cancer. Okay, its main role is as the frontline defense mechanism against the sort of stresses and processes that would bring about cancer. And so it's no surprise when I tell you that it's actually the most mutated protein in cancer. When unfortunately cancer does develop, loss of this particular guardian of the genome, this protein P53, is necessary for the progression of that cancer. One thing that's quite amazing that we can do in the lab, not in the clinic, but in a laboratory setting, is take a cancer that's lost P53 and bring it back. We restore the guardian of the genome, and invariably, and this has been reproduced time and again, cancers go away. So the take-home message is that we have a potential Achilles heel for cancer, and it's within normal bodies preventing cancer on a daily basis. But unfortunately, despite decades of understanding that this protein is doing this critical role, we don't have drugs that directly target it. And that's because it's one of these proteins that is referred to as undruggable. And I take great insult with that definition because it's something we need to change. Now, the vision for my lab is that we can boost proteins like P53 using epigenetics to bring back, in a cancer situation, the body's natural anti-cancer defense mechanisms. And so there's an illustration on the left of some big complex data like the sort Chris was talking about. I don't want to drill into the complexity here. That's my job to understand that. But we can use these very sophisticated epigenetic understandings of proteins like P53. If we understand how it works, can we use epigenetics to make it work better? And this is a slide where I'm showing an illustration. It's a microscopy slide where we're doing exactly that. On the left, you have cells which are growing under stress. P53 is trying to do its job, but not quite able to achieve its role as preventing these cells from continuing to grow. On the right, we've synergistically targeted P53 as well as epigenetic processes to kill all of the cells in this dish, which is what we want to achieve. In essence, what I'm showing you here is that we've used epigenetics to boost the function of this critical tumor suppressor protein, this defense against cancer. We can do this in the laboratory. Our dream is really to put this into practice within the clinic for patients. This is the last slide that I'll have. I wanted to really detail the foundational pillars of medical research. You have fundamental research, which concerns itself with discoveries in the dish, in the lab environment. You have translational research, which is so important to make sure those discoveries are robust. And then, of course, you have clinical research, which drives home the impact of those discoveries. And you'll notice there's a missing part in the center, and I think that that's this emerging pillar of medical research, which is community, which is really so important for the work that we do. I'll give you 2 examples of where we use community to strengthen our research. One is where we take this community lens, this understanding of what we do, which often is a bit opaque, and we try and improve our ability to tell the stories about the research that we do using community involvement. So they help us shape the messages that we have. They obviously provide incredibly important philanthropic capacity for doing what we do in the lab. That's an emerging thing which is so important for our research. And frequently these people with lived experience of the diseases that we work on are so important in actually guiding the work that we do. In many cases, even experimentally. You ask any survivor or person with lived experience in cancer how they would do the research, and I bet you they'll have very critical and interesting lessons to tell us. And with that, I'll end my talk, and I'm very, very grateful for the audience today. It's been absolutely amazing to come and talk to you, and yeah, just very, very valuable. Thank you. That's a bit preemptive. We've got some question time, apparently. There is just one announcement I would like to make, if that's okay. May 13, is that right, Mitzi? The SAiGENCI annual public lecture being held in the Scott Theatre. I'm sure Mitzi can get some information disseminated to anyone if you haven't received it in your emails already. May 13, 5 o'clock, 5:30, Scott Theatre, May 13.
So, when you talk about epigenetics and the other parts of your work, then how focused is that on cancer? And I presume that a lot of that is applicable to other diseases as well, other conditions. So how much is that you focus purely on cancer, or do you essentially, well, I focus on epigenetics, which happens to be in the context of cancer, and we're doing it useful for any other context as well?
I've never met you before, and I didn't ask you to ask that question beforehand, just for the record. But it is true. So everything we do, all of the lessons that we learn in our particular environments or experimental settings are communicable to some extent, some greater than others, to every part of biology. We call ourselves systems biologists because we study systems. And with the sort of tools and techniques and cutting-edge world-class platforms that we have, we can communicate across different systems. The fundamental lessons we learn are important and applicable for every, to some extent, every disease that you might want to know more about. P53 is a type of protein where we can learn lessons and apply across many other different epigenetic regulators. So developmental disorders, not just cancer. Absolutely.
Did you bring some P53 pills with you today? There's a few of us here who'd like a few. My serious question is, how will that be delivered? How will you actually create that in terms of what's the point of from knowledge to understanding to delivery? Thanks.
So that's where I'll pick up on the notion that we are a cancer institute based in the greater ecosystem of the University of Adelaide University. And we actually recruited a young fellow called Michael Roy,
who has developed this new technology of working out how to drug the undruggable with a technology called PROTAC, which degrades aberrant control mechanisms. Sometimes we can hit a drug directly and put the key into the lock and turn it off, but oftentimes those keys are not available, so there's been a whole new area of chemistry to be able to drug these new therapies. And for example, we've recruited a gentleman to build that technology in South Australia to develop those new therapies. So it's a chemistry question. There are major advances in chemistry, and we're working with the chemists.
I have a question. How transferable is this research to other forms of cancer, for example, multiple myeloma, leukemia, etc.?
That's actually quite easy to answer. We have set ourselves up as a cancer agnostic institute. So all those programs we described are relevant for all cancers, be they a solid tumour, something that comes from, say, the prostate, the breast, the lung, as well as the blood-borne cancers like myeloma that Suzanne's working on and other leukemias and things like that. So what is the difference between those cancers is the microenvironment. How do they live in their different blood systems for the blood cancers? How does a prostate cancer live in the prostate, grow in the prostate, but then go to the bones? So that's a microenvironment question that differentiates them, and some of our work is overcoming how does the soil support the cancer, and how can we block the soil from supporting the seed in the cancer in the soil. Sorry, the seed, which is the cancer in the soil.
Very, very interesting, actually, and I'm not into technology, by the way. But my wife had breast cancer about 18 years ago, and with a positive thought, fortunately, she survived, and now she's free of cancer. And it amazed me that, I don't know how that happened, is the fact that you go to somebody about it and things like this. She didn't take tablets or anything like that, but she recovered and completely, and she kept a positive attitude towards it. Does that help?
So I'll dissect that in a couple of different ways. The best way to cure cancer is to prevent it, not smoking, for example, for lung cancer, or go through your screening practices to diagnose it early when it's contained and hasn't spread into the local environment of the breast and moved to other organs. So early diagnosis was key. But what you allude to, is there something where the body itself can help augment its anti-cancer fighting properties? And the answer is a strong yes. So a good healthy diet paired with exercise. So getting your weight under control, exercise does actually boost a whole series of anti-inflammatory immune systems. Just like it fights viruses and bacteria, it can also fight cancer, and we know that is better in healthy organs, healthy humans. But also there is something to the neural axis generating anti-cancer proteins in the body as well. So diet and exercise and a healthy lifestyle is key to preventing as well as containing it if it is actually present.
Hello, thank you, Professor Luke. The comment that I hear made very often amongst us mere males is that we're likely to either die of prostate cancer or with it. And I have lost count of the number of friends and acquaintances who are dealing with prostate cancer in recent times. I'm wondering, in terms of the work and the research that you are doing, what are some of the indications of change that you hope to be able to bring about to people who are mature age males and dealing with this? Is that too general a question?
No, it's not. And that actually, prostate cancer is an example of how we're trying to manage and decrease the death rate from blank cancer, prostate being one of them. And so early diagnosis, so one of the projects I've done with the clinical trials and the thousands of specimens I've generated from around the world is brought them into the data into one database and saying, what is the genetic profile of the gentleman that has the low risk cancer that is diagnosed with and doesn't die from?
versus the aggressive one where some people do die of prostate cancer. So we're trying to actually get a feel for what is the genetic makeup of the people and identify those who have the high risk profile and identify them through screening programs and not waffle around, is the PSA up or down, do we need to do an MRI, but really target and find that genetic profile of those at high risk for the cancer beyond the BRCA mutation, for example. So that's a process we're trying to do using artificial intelligence to find those risk profiles. But we're developing new strategies to overcome the resistance mechanisms, P53, NF-kappa B, other ones, and we are developing a drug against that makes the hormone therapies more effective. And the clinical trials, I'll take a moment to brag. In the 1990s, the 8-year survival was 25%. And I've been leading a couple of trials that actually have changed the way we treat prostate cancer. And we now have men alive with prostate cancer, with metastatic disease, at 8 years. So it used to be 25% alive with metastatic cancer at 8 years. We're now up to 50% over a period of 15 years with these new therapies. So we're doing it from trying to prevent, early detection, as well as improve the therapies we have.
Can I ask one last question? We often hear, usually in the press, that the incidence of certain types of cancers is increasing. And in fact, it was something in the Australian a couple of days ago. Is it possible that the introduction through your science into individual cells in a sufferer from a certain cancer, can that be inherited so the likelihood of that cancer appearing in the offspring of either the mother or the egg or the sperm of the male, that will slowly reduce the incidence of incidence of cancer? Is it inheritable?
So you go with that. During my PhD, I worked quite a bit on the inheritance of epigenetic information. This concept that you would have one generation acquiring epigenetic marks that then get transferred via the sperm or the egg to the next generation. We do know that this is possible because we've got fantastic models of where this has occurred. At the end of the day, it's extremely hard without very sophisticated research to track how this would occur in a real world situation with humans. We do think it's possible, but the literature here is a little bit murky and clearly needs to improve, especially with respect to cancer. Separating genetic from epigenetic changes is extremely difficult, and that's really what we're trying to do in many respects.
And I'll just pick up on that. That's so why people know of the BRCA gene, the Angelina Jolie gene. That's an example of an extreme mutation that puts people at risk of breast, prostate, ovarian, and pancreatic cancer. There are others that are a bit more subtle that put people at risk but not guaranteed to develop the cancer. And then there's the environmental aspect. So it's a spectrum of how much the genetic inheritability is. But what we should all do is work on the diet and exercise, if I'm going to harp on that again, to diminish our risk is the most powerful way, the most powerful tool we have to diminish the risk of cancer.
Well, thank you very much, Professor Sweeney and Dr. Isbell. I suspect there was no one in the room who doesn't have cancer or doesn't know. As our population grows older, it's more likely. So, would you put your hands together and thank you?